Characterization of bacterial culture in agar lands. Agar lands are used for storing pure cultures for a moderately long term. There is minimal risk of contamination or of losing the culture due to the medium drying out, because the small volume of air inside the tube and narrow opening combine to limit water loss and expose the outside air, including dust and outer or other particles. With that, we will characterize the Pseudomonas aeruginosa grown in a agar-land culture. For its brief rundown of its morphological characteristics, we have its shape, which is a slender rod-shaped bacterium. Size. The size of the P. aeruginosa is about 1.5 to 3 micrometers by 0.5 micrometer. The arrangement of cells. Pseudomonas aeruginosa is arranged singly or in pairs. For the motility, it is an actively motile bacterium. For its flagella, it is flagellated bacterium with a polar flagellum arranged in an amphitrechous manner. Spores. The Pseudomonas aeruginosa is a non-sporing bacterium. Capsule. P. aeruginosa is a non-capsulated bacterium, but some strains possess a slime layer. The gram-strain reaction. Pseudomonas aeruginosa is a gram-negative bacterium. To start, for the amount of growth, Pseudomonas aeruginosa is a common, free-living, gram-negative bacterium that can cause significant disease as an opportunistic pathogen. Rapid growth, facile genetics, and a large suite of virulence-related phenotypes make the P. aeruginosa a common model organism to study gram-negative opportunistic pathogens in basic microbiology. Hence, it is abundant in terms of the amount of growth. And for the color of the colony, as observed in a slant agar culture, the color of the colony is red because of the presence of the lactose, glucose, and sucrose. The P. aeruginosa is a lactose fermenter, responsible for the color red. In a McConkey agar, on the other hand, the colonies of the Pseudomonas aeruginosa are colorless due to the lack of lactose fermentation, which is of great importance in differentiating P. aeruginosa from other bacteria present in the specimen, especially from a gram-positive bacteria, examples the E. coli and the Klebsiella species, which are lactose fermenters and gives pink-colored colonies on a McConkey agar medium. And to add, for the pigment production in the culture of the Pseudomonas aeruginosa. In the culture medium, the P. aeruginosa produces mainly 4 types of pigments which diffuse out into the surrounding medium and imparts a characteristic color to the medium, indicating the suspected growth of the Pseudomonas aeruginosa. Mainly, the pyocyanin, which is bluish to green color, the fluorescein, which contributes to the greenish to yellow, the pyorubicin, reddish to brown, pyomelanin, brown to black. Of the 4 types of pigments, only the pyocyanin is important for the diagnostic point of view, as pyocyanin is the only pigment that is not produced by any other species of Pseudomonas, but only by the Pseudomonas aeruginosa. It is imparting characteristic color to the medium, representing Pseudomonas aeruginosa growth in the medium. And followed by the consistency. Viewed by a transmitted light, the colonies were smooth, convex, opaque, glistening, entire, and pyocyanogenic. They were butyrous to the touch and produce a uniform turbidity when suspended in a 0.85% solution of sodium chloride. For the optical characteristics, the colonies of the original culture of the P. aeruginosa, or the ATCC number 10145, had a raised, opaque, glistening center bordered by a depressed, less opaque, dull, concentric ring. The colony mass tapered gradually from the outer edge of this ring to an irregular, translucent margin. And for the form of the P. aeruginosa. The form of the P. aeruginosa in a slant culture medium is filiform or uniform along the line of the inoculation. And for the odor, in an ordinary medium, the Pseudomonas aeruginosa produces a characteristic sweetish odor or a distinctive musty or earthy smell in the medium. Pseudomonas aeruginosa grown in the media is also characterized by a grape-like smell. Being a non-fermenter, the bacterium is also associated with acid production in culture rather than gases, which are commonly associated with fermenting bacteria. And lastly, for the medium change, there is none color change.
What is slanting, or agar slants? Agar slants are created by bringing agar to the boiling point and pouring it into a test tube. Before the agar cools and solidifies, the test tube is set on its side. And once the agar is cooled, the test tube can be stored upright and the agar inside has a slanted appearance. When examining agar slant culture, some of the things we look for is the amount of growth the culture has had, ranging from no growth to abundant growth. The next that we look for is the form they take while growing in the culture. It differs from bacteria to bacteria, and some bacteria, like the Serratia marcescens, have a filiform, or a thread-like appearance. Other bacteria can have a beaded appearance, a papillate, a villus, meaning it has lots of villi, and some bacteria can also be arborescent, or tree-like. Lastly, we look for liquefaction, which is the reduction of a gelatin culture medium to the liquid state by enzymes produced by bacteria in the culture, and liquefaction can take different forms depending on the bacteria. The first that we will be looking at when examining agar slant culture is the amount of growth. As said previously, it can range from no growth to scanty, moderate, and abundant. Starting from the bottom, we have none. When there is no growth in the culture, there could have been an error during the process and might need to repeat the experiment. And simple errors, such as not letting your inoculating loop or needle cool properly, can affect the culture in such a way as to leave you with no results in your agar slant. Scanty. When cultured, the Bacillus hoffmanni, or the pseudo-diphtheria bacillus, only has a scanty amount of growth. The culture, in other words, has had small or little growth during the culture period. The next one is abundant. Here we see the Sabouraud agar slant culture test, and it is used when fungal overgrowth of the oral tissues is suspected, and it is used to confirm the diagnosis of a fungal infection. Notice how the fungal infection has proliferated to such an extent that you can barely see the surface of the agar. This is classified as abundant. The characterization that we will be examining next is the form the bacteria grows into. The first form we will look at is the filiform, or thread-like form, and the examples of this is the agar slant tube cultures of Serratia marcescens, which is the left side colored red, and I'm going to try my best at pronouncing this one, Cochuria rhizophylla. On the right, we have Micrococcus luteus. Also, Bacillus subtilis has a thread-like form, and most often, the bacteria grows on the area that has been inoculated, hence why we see a streak thread pattern or form. Citrobacter and Salmonella, with the subspecies Arizonae, also have filiform, or a thread-like growth following the trail of inoculation on the TSI slant. TSI stands for the triple sugar iron test. The next form is a beaded form. It's called beaded due to it looking like beads attached to each other or in very close proximity. An example of this is the Bacillus hoffmanni, as we saw from earlier. Not only has a small amount of growth while it's being cultured, it also takes the form of beads connected or very close to each other. Another example of a beaded form is the Streptococcus lactis. The next forms that we look at is papillate, which has a nipple-like resemblance. Next one is villus form, and they're called villus because they are covered with villi. The next one is arborescent, and they resemble tree branches, or trees themselves. The next characteristic we will look at is liquefaction. Some bacteria cause a liquefaction, which spreads across the whole upper surface of the gelatin and continues gradually downward. The bacteria turns the one solid agar to a liquid, and in the process, shape the agar in different patterns. These patterns are criteriform, napiform, infundibule, saccate, and stratiform. The first one that we shall look at is criteriform, and an example of this is the cholera spirillum, and it is categorized as a criteriform due to the surface area being turned into a saucer shape. The next pattern is napiform, and is characterized as having a bulbous shape, or a bulb-like shaped liquefaction at its surface. The next form, or pattern, is characterized as being funnel-shaped on its surface, and this is called infundibule or infundibulum. The next form or pattern is characterized as elongated and tubular, stretching nearly all the way down to the bottom of the tube, and this is called saccate. And the last form or pattern is characterized as a complete liquefaction of the upper half of the medium, as shown in the example, and this is called stratiform.